Uh, I'm Dave Long. I'm the co-president of the Marin Chapter of CNPS. I want to welcome you to our January meeting. And I imagine most of you know the protocol. We would ask you to stay muted during the presentation and encourage you to put any questions as you think of them in the chat. And then there will be an opportunity at the end of the presentation to ask questions individually and to unmute and ask them yourselves. We have the honor and pleasure to have Miranda Melen with us tonight to talk about invasion stink, the response of Stinkwort, Dittrichia graveolens to competition and disturbance and its seed bank dynamics. So I think we are going to learn about how invasives interact with natives and hopefully some more information about how to control them. Miranda is a Ph.D. candidate specializing in plant invasion biology in the Department of Ecology and Evolutionary Biology at UC Santa Cruz. She's the recipient of that means the Marin Chapter CNPS 2023 Howard-Kohn Memorial Scholarship to support her research, which focuses on understanding of the spreading potential of invasive species and the underlying mechanisms that drive their invasion. And her focus, as I mentioned specifically, is on Stinkwort and its competition dynamics and seed bank persistence. She has a background also in restoration, in vegetation management, and plant conservation and teaching, and aspires to teach at the university level and has a great interest in engaging and supporting underrepresented students in the STEM fields. So it's with great pleasure that I turn the meeting over to Miranda Melen.
Thank you very much. Hello, everyone. As you said, my name is Miranda Melen, and I'm a PhD candidate in Ingrid Parker's lab at UC Santa Cruz. Today, I'm going to be talking about the response of Dittrichia graveolens, specifically to competition and disturbance, as well as its seed bank dynamics. Here, I'm presenting research on behalf of what we call Team Dittrichia, which is comprised of Ingrid Parker, Nikki Lustenhauer, and myself, in addition to a really amazing group of undergrads that we've had the pleasure of working with over the past few years at UC Santa Cruz. In this talk, I'm going to provide first an overview of Stinkwort biology, and then I'm going to go into the response to disturbance and competition and what we know so far about its seed bank dynamics. And then at the end, I'm going to give a quick plug for an upcoming Stinkwort workshop that is coming up next month in February. First, let's review together what we know about Stinkwort biology. Dittrichia graveolens, or Stinkwort, is an annual in the Asteraceae family, and it hails from the Mediterranean basin in Europe. Stinkwort has invaded Australia, South Africa, South America, and North America. In its native range, it is found in a variety of disturbed environments, whereas in its invaded range, it's first found primarily along roadsides, which I'm sure many of you are quite familiar with seeing as you drive around Marin County or other places in the Bay Area where we see it along the roadsides, whether they're highways or verges along other areas. Here's a map that shows Stinkwort's native range in green. You can see that it mainly hugs the Mediterranean Sea, but it also extends west into Asia. The orange we show is the expanded range. And in Europe, that means that the northern expansion is more recent and still considered part of the native range. But in North America, South Africa, as well as Australia and New Zealand, Stinkwort is classified as invasive. In its introduced range in California, Stinkwort was first identified in Santa Clara County in 1984. As of 2020, it was found in 79% of California counties. This map here is adapted from Brownsey et al. 2013, and it shows the progression of Stinkwort through time in California. The black polygon is Santa Clara County, and as the colors move from blue to green and dark to light, you see more recent spread events throughout the state. The bright or light green polygons that are more along the edge are more recent invasions of Stinkwort, and that is in comparison to this core region that we call the core area or the core region in Santa Clara County, where it has been observed for, it's 2024, so 40 years. It spreads primarily along roadsides, but has been found in plant communities away from roads, which is concerning, since little is known about its invasion potential in these less disturbed environments. This weed risk assessment graph from the USDA shows Stinkwort to have higher establishment spread potential, coupled with a moderate impact potential. This places Stinkwort, the black square, where I have the giant red arrow, in the red X region of this graph, identifying it as a high-risk major invader. Stinkwort is classified as a noxious species in California because it produces poor fodder for animals, can cause milk tainting, and harmful when ingested by livestock due to the barbed pappus on the seeds. In addition, exposure to the oils can cause contact dermatitis for some humans. Hopefully not many of you have experienced that, but I certainly have had undergrads come back to me saying that they need to have different clothing on when they're working with the species. Stinkwort is a late season annual, which means its phenology is offset from many of the local species in California. I think this is really cool, but it can cause some concerns for us as we're dealing with an invasive. Although Stinkwort readily germinates after rain events, it then spends much of its early months as a small rosette, and it only invests in above ground biomass in June when it begins to bolt and produce secondary growth. By July and August, it starts to produce buds so that its main flowering season is from August through September, with seeds lasting on adults through December. Stinkwort germinates after heavy rainfall events, but it can be really difficult to identify in its seedling form. And this makes it challenging to control early in the season. Here's some photos that I've taken or found that show the seeds up close, as well as the cotyledons and first true leaves, which have been very difficult to identify in the field. Seedlings have hairy leaves that are teardrop shaped, and the often cryptic rosettes can stay inconspicuous until summer heat cues bolting. That's when leaves are larger and closer together during this stage, and the rosettes can be small, like the above photo, or more dense, like the lower photo. We suspect that differences in germination timing and moisture availability during this stage dictates the size and density of the rosette. But the bottom line is to look for a variety of rosette forms. During the summer, when native plants have senesced or gone dormant, Stinkwort starts to produce secondary growth and reduces its leaf size. Leaves and stems are hairy and sticky during this stage of growth. And the scent of Stinkwort is very strong when the plant is disturbed during this stage. Some people think that the smell of Stinkwort is pleasant. I am not in that camp. This is also the time when the classic Christmas tree architecture takes shape in a more robust individual, like the one on the right. Buds tend to show in late July or August, depending on the summer heat. Yellow flower heads begin to form in late summer, although I noticed in Santa Cruz this last fall that flowers didn't show until October. Buds, flowers, and fruits can be on the same plant at the same time throughout the fall. Stinkwort fruits are wind dispersed and continue being produced through December. The pappus allows the seeds to ride the winds and stick to socks, pants, weed whackers, and vehicles, helping to transport the seeds to new locations. Often when I leave a site, I end up dusting down my shoes and pants to prevent seeds from hitchhiking. Stinkwort has a good root system while it's in its rosette form and develops a taproot during the bolting stage, which may be a key adaptation to California's soil conditions. More studies are needed to understand the resource use of stinkwort and how it manages growth during our dry summers. We see stinkwort growing in many different types of environments. So here I have 4 different pictures. This includes pavement cracks, dry, salty flats when I'm out at the baylands around San Francisco Bay, invaded grasslands, such as up in the Mount Hamilton area, and then, of course, rocky roadsides. Here is a photo of Lexington Reservoir in Santa Clara County, which experiences disturbance along the barren lake shore. And the green Christmas tree-like plants, yes, indeed, are stinkwort. So this is what I qualify as a land manager's nightmare. I think you would agree. So now we've reviewed the biology of stinkwort. Let's jump into the research portion of the talk. Next, I'm going to talk about disturbance and competition. Invasive species are a major resource management problem, and we all spend a lot of effort controlling them. Yet only a small proportion of introduced species will actually become invasive and cause ecological impacts. Risk assessment includes evaluating which habitats are vulnerable to invasion by a species, but also which traits may make that species more likely to invade those habitats. Yet traits may evolve. Therefore, rapid evolution makes it harder to do risk assessment because of potential change. Transportation corridors play an essential role in the establishment of introduced plants. Vehicle traffic moves plant propagules along roadways, speeding up dispersal rates or establishing new roadside populations. And road construction and maintenance makes environments with poor soil fertility and low competition. Species that grow along these roadsides tend to have the classic rural life history: high fecundity, short generation time, and long distance dispersal traits. Stinkwort is found along roadsides all over California, but we are especially worried about it because it is starting to appear spreading away from roads. Where and why this is happening is a critical question we're trying to answer. Here I have a picture of Dittrichia growing along a roadside. You can see the tire marks from a vehicle that has moved through this gravelly area. And then this is a picture from a springtime where we have a grassy hillside, that's the green, and then the old skeletons of Dittrichia, demonstrating that there's an extent population that is going up this hillside. And this is in the Mount Hamilton range. I want you to recall that in its native range, stinkwort is found in a variety of disturbed environments and has recently started spreading north in Europe. Remember that map that I showed you where we've got green from where the native range is, and then orange is that the new populations that are spreading northward. In its invaded range, stinkwort is first found along roadsides, and then it moves into other disturbed environments. So our first question to address was, why does stinkwort thrive in these disturbed environments? We started with the premise that perhaps stinkwort is promoted by disturbance reducing competition. We did a field experiment with study plots in an invaded annual grassland in California. For this experiment, we had 4 disturbance treatments: grassland control, competitor removal, raking, and raking and clipping. For the grassland control treatment, we did not disturb the background vegetation and instead just planted in stinkwort seedlings right into the vegetation that was growing. In the competitor removal plots, we hoed each plot to disturb the soil and remove above and below ground competition. And then we planted in stinkwort seedlings. In the raking plots, we raked away thatch before planting stinkwort into the vegetation that was already there. And in the raking and clipping plots, we raked away the thatch before planting Dittrichia. And then we went back during the growing season and clipped non-stinkwort vegetation low to control above ground competition. In each plot, when we planted stinkwort, we planted in 16 seedlings and monitored growth. I would have loved to have grabbed an earlier stage and had seeds in those plots, but to be conservative and to be a mindful scientist, rather than putting seeds out into the field, we took seeds, started them in Petri dishes at UCSC, and then once they had germinated, we transferred them into pots, then transported those pots to our field site and transplanted them into the ground. We allowed them to grow over their growing season, and when a stinkwort plant began producing buds, we harvested the above ground biomass. Again, being very mindful to not release seeds into the population, not to release seeds into the environment, either by having seeds from our initial experiment or by producing seeds from the plants that we had planted. When we compare stinkwort biomass by treatment, we see a difference when we disturb the soil and remove competitors. In fact, stinkwort had 17 times greater biomass in competitor removal plots where soil had been disturbed and competitors removed compared to all the other treatments. This study supports that as a ruderal species, stinkwort is a poor competitor and may be associated with these less than desirable disturbed environments because competition is low. Now, we have more studies in progress to further explore these patterns, but this result helped us think more deeply about the role of competition and whether evolution may contribute to stinkwort adapting to more competitive environments. So think back to when I was talking about how traits can evolve and the concerns around that relating to rapid evolution. So the pattern of spread in California, coupled with stinkwort's rural life history, made us ask, what happens when a roadside weed moves into vegetation where it deals with competition? Stinkwort is a natural choice for testing the role of adaptive evolution in invasions. And we wanted to look at multiple aspects of invasion where evolution may be driving change. We were curious to know if stinkwort populations already experiencing competition germinated faster or flowered sooner, maybe survived better, or had higher biomass than their roadside counterparts. Here, we tested whether stinkwort populations have evolved during their spread away from roads in California. And we did this we tested for differences in germination behavior. We used a greenhouse experiment to test for response to competition in field soils. And in a field experiment in an annual grassland, we compared genotypes from roadside and vegetated sites in their flowering time and their response to release from competition. In fall of 2020, we located 8 pairs of stinkwort populations in Santa Clara County by finding a population growing in vegetation and pairing it to the nearest roadside population. There are several reasons why we picked Santa Clara County. First of all, it was 2020 and I live in Santa Clara County. The second reason was because, if you recall, Santa Clara County is where Dittrichia was first identified 40 years ago. So in this core area, Stinkwort has been around the longest. And so we were thinking that the potential for finding local adaptation would be more likely in a core region rather than on some invading front going up the Sierra Nevada foothills or along the north or south boundaries of Stinkwort as it further expands in California. Here's a map with each location. And then as an example, there are two photos that show a Stinkwort population growing in a plant community and a Stinkwort population growing along a roadside. Just so you have a good visual for what do I mean by a roadside population, where you can see there is Dittrichia, there's Stinkwort, and then there is the road going past it. And then in the above photo that's labeled vegetated, you have Stinkwort growing in senesced, probably a recently mowed area that is a vegetated plant community. And yet Dittrichia is growing there just fine. When we compared source habitats, we found that roadside habitats had higher amounts of bare ground and 3 times less cover of resident plants compared to vegetated habitats. Now, overall, resident plant species at all sites were predominantly non-native annuals in all of these surveys. And species richness was the same between habitat types, so these roadside and vegetated habitats. After completing these plant community surveys, we collected seeds from each population to use in our experiments. First, we asked whether there was a difference in germination between seeds from populations in roadside and vegetated sites. We wanted to understand if seeds from vegetated populations had evolved to germinate faster to outcompete resident vegetation. Seeds that germinate faster can gain access to more light, water, and soil nutrients, which can provide an early advantage in a more competitive environment. In this experiment, we placed 10 seeds from each source habitat in Petri dishes with one of 3 treatments. We had filter paper, engineered fill, and topsoil. And as an aside, filter paper was a good control just to help us understand whether these seeds were viable and kind of the baseline of ideal germination conditions and what would be the viability of those seeds. We had a great source of engineered fill, which is very much similar to the conditions that are along roadsides as they lay out a roadbed. So this is a soil that has been engineered by humans for purposes of construction. And then we had topsoil, which is a great proxy for the type of soil that has more nutrition for that you would find a plant community. So these seeds are growing in these different treatments, and we allowed them to germinate and checked them each day to count the number of germinated seeds to assess the rate of germination. Okay, so seeds originating from vegetated habitats consistently had a slightly reduced probability of germination compared to seeds originating from roadside habitats. And we saw this in our filter paper results, engineered fill, as well as topsoil. What this means is that the seeds from populations experiencing competition actually had a lower proportion of germination, which is counter to what we expected if these populations were adapted to competition. We next wanted to explore how Stinkwort responds to competition with other species. We selected 2 non-native grasses as competitors for a controlled competition experiment in the greenhouse. We used Bromus hordeaceus and Festuca perennis because they were both identified in our plant community surveys and are commonly found in California annual grasslands. We grew stinkwort in 3 competition treatments: alone, with festuca, and with bromus. And then after 4 months, we harvested above ground biomass. Before we get to looking at the data, I'm giving you an example graph to help us interpret what's on the next slide. We use something called log response ratio as a way to assess the growth response of stinkwort to the absence and presence of competition. This means we took the natural log of the biomass from alone plants and divided it by the biomass of the plants experiencing competition. A negative LRR, log response ratio, when we have a negative LRR value on the y-axis, it means that grass had a negative effect on stinkwort growth. Now, let's look at the slope of the line in this example data. A positive slope means that seeds originating from vegetated populations grew better under competitive conditions than roadside. This positive slope is what we would expect if stinkwort has adapted to competition. Now let's look at our data. Okay, we're going to walk through this. First, we see that indeed, stinkwort was strongly affected by competition with non-native grasses, regardless of the source habitat, because the y-axis is negative. When we look at bromus versus festuca, we can see that festuca is a stronger competitor against stinkwort compared to bromus, because the data are all shifted down to a more negative place in the graph. But when we look at the slope between the roadside and vegetated source habitats, we see a mixture of outcomes, and that's saying it nicely. It's all hibbly-jibbly. This amounts to stinkwort demonstrating a lack of competitive ability, but showing a clear signal that it does not do well in competitive environments. Okay, next, we took our question about competitive ability and tested the seed source habitats under field conditions. We used 2 treatments here for this experiment: competitor removal, where we hoed the plots to remove above and below ground competition before putting in Dittrichia, and a grassland control, where we allowed the grassland to grow up around the plants. We placed 16 stinkwort seedlings in each treatment plot and harvested above ground biomass when flower buds formed on a plant. We examined the success of stinkwort growing under competitive and non-competitive conditions. We measured survival to bud and biomass. And regardless of the habitat source, plants from roadside and vegetated sites did not differ. Finally, we also assessed flowering time between the 2 source habitats and treatments. We found that there was no difference between source habitats. We did see a treatment effect where both source habitats delayed their flowering in the competition treatment, but there was no genetic differentiation. So the conclusions of this part of the study is that we know that evolution in introduced species can be an important driver of invasion. And here my team looked at germination, we looked at competition in a greenhouse and competition in a California annual grassland to really try and triangulate and look at this question from different angles. But we found no evidence that stinkwort populations in roadside and vegetated source habitats have undergone genetic differentiation that could be considered adaptive. So the takeaway is that evolution has not increased stinkwort's invasion success in California grasslands. Now I'd like to talk to you about a 5-year ongoing study that we are doing looking at seed bank dynamics. We are interested in how Stinkwort seed germination, dormancy, and viability affect the longevity of the seed bank. Oftentimes, when we're working with invasive species, we're thinking about how are we controlling or removing a particular invasive species from an area. And we're well aware that there is a seed bank that we have to keep coming back to a certain area for a certain amount of time to get anything that has germinated. We don't really have a clear understanding of what the longevity or dormancy rate is for Dittrichia. We are also interested in whether there is reduced dormancy in populations from the range edge in California. So this is another example where we're looking at core versus edge types of populations, where there may be a difference in how dormancy plays out in a population that has been around for 40 years compared to dormancy that is on the leading edge of an invasion, where it may be advantageous to break dormancy sooner so that you can get to the next population location and put down seed, grow up plants, and start the successive cycle of getting your annual reproductive cycle going. So to answer these questions, we've been conducting experiments in a common garden or mesocosm, as well as in the lab and field. The mesocosm and the field experiments are 5-year experiments that are ongoing. So I just have preliminary data to show you. Back in 2018, we collected seeds from stinkwort populations in 2 areas of California. So we sampled from Santa Clara County, as you recall, which is considered the core region since stinkwort has been here for at least 40 years. And we also collected seeds from the edge of the invading front along the Sierra Nevada foothills, where populations are more recent to the area. And we were able to identify corridors, 4 of them, that go up into the Sierra Nevada foothills where we could collect Dittrichia. We collected from 4 populations in the core region and 4 populations in the edge region. And then seeds were grown in a greenhouse for 2 generations to control for maternal effects. To assess germination, we developed a 5-year mesocosm experiment at UC Santa Cruz. And this picture here shows, I've got 2, you can think of them as 2 raised beds. Within the raised beds are numerous pots, and in each pot, there are seeds from a particular individual, from a particular population on either the core or edge populations where we collected. So in this experiment, we placed 200 seeds per pot in these open pots that are outside where they receive natural light and weather conditions. We check the pots very regularly for germination and count seedlings that emerge. So all of the rain that we have had recently, we've been checking, and we have seen when we have germinations occur, and we're making germination counts as we go through those rainy seasons and then throughout the year to make sure that we don't have any germinations that go unaccounted for. Here is the top right picture, which is a top down of one of the raised beds, has kind of my first nightmare, where we looked at the pots after the first rain in year 1, and there was green all over the place. And I realized I needed to get through these pots pretty quickly to count all of the germinations before we started having seedlings crowding and outcompeting each other. The picture in the lower left shows a close-up of one pot where you can see that the seedlings are really crammed together. But I had amazing undergrads like Josie here who were able to help me go through each of these pots and systematically pluck and cut every seedling, count them, and come up with a good method for doing data analysis here. So here I'm showing the first 2 rainy seasons with rainfall on the right axis with purple columns and germination proportion on the left axis with green dots. We saw high germination in the first rainy season, with additional germination occurring in the second rainy season. And we're still collecting data from the current rainy season, but have seen additional germination occurring 3 years after the seeds were placed in the pots. I don't have data to show on this graph, but they're still coming up, which is surprising but expected. So these seeds were placed on the soil surface, which mimics how Stinkwort seed settles on bare ground. Here we are visualizing the seed fates for year 1 of this experiment using something called a Sankey diagram. And we can see that in the first rainy season, 85% of the seeds germinated and 15% remained in the seed bank. When we calculate the viability of those seeds based on other viability experiments we've done, we estimate that about 12% of the seed bank is still viable. So this is what's happening on the soil surface. But we all know that seeds get moved around and that their behavior can be really different when they're buried under the surface. So next, we asked how Stinkwort germination responds to different seed depths. We placed seeds at 6 different seed depths in moist sand and watched for germination. So here is kind of like an infographic where I have gradations of a seed sitting at 0 centimeters, but we also had 5 other placements of seeds at 0.1, 0.5, 1, 1.5, and 2 centimeters underneath that soil surface. We had 10 seeds per tube, and we moistened the sand with DI water as we were watching to ensure that we were getting all of the germination. We found an interesting but expected pattern. When the seeds are on the surface, we see around 70% germination, but the germination rate drops to about 45% with only 0.1 centimeter of sand covering the seeds. And then by the time you have 1 centimeter of sand on the seeds, germination rates drop to under 20%. So we know from this that those buried seeds are not germinating, which means it is critical to understand the dynamics of survival for those buried seeds. All right, so we've seen what happens in a mesocosm when seeds are placed on the surface, and we've played around with the seed depth to better understand germination when seeds are covered. Let's move to the field and check in on a 5-year seed burial experiment. So the setup of this experiment uses the same seed source as the open pot mesocosm, replicated in 4 time intervals. I have a few pictures here from the field showing how these seed bags are buried. I say seed bags, but I think of them more as seed sleeves. They were long pieces of fabric that were sewn to have cells, and the seeds were placed in each of the cells. Each of those cells, the seeds came from a particular population, a particular individual that was either in the core or the edge, and everything was randomized. And in each time interval, that is a replicate. So in year 1, we're able to pull up a set of seed bags, and we can have those same individuals in year 2, year 3, and we skip year 4 and have year 5. So in the upper right photo, there are 4 sets of seed bags. And so far we've pulled up the first and the second set. Each year of harvest, we pull up 1 set of seed bags, and then we go through the meticulous duty of counting how many seeds remain ungerminated in each of those cells. We then take those seeds and we place them in a petri dish, and we dose them with gibberellic acid which is a growth hormone that breaks dormancy. So this helps us quickly assess of those seeds in each of those cells, which ones were really secretly dormant, but just needed a little hormone boost to germinate. We then count the number of seeds that germinate, and any remaining seeds are treated with tetrazolium and visualized under a microscope. Now, tetrazolium turns living cells pink. So a seed that is pink means that it's still dormant, even though we gave it gibberellic acid. So this is kind of that backup, a backup test to make sure that we're really understanding whether a seed is alive or dead. But if a seed is blotchy pink, where there is not, where the pink is uneven, and so there is some dead tissue and some alive tissue, or if the seed is white or black, it means that the seed has met its death. So the bottom two, or the bottom four pictures, I have an a dirty seed bag, a seed bag that we pulled up from the ground. And then I have a petri dish that shows after we've dosed it with gibberellic acid and let it germinate, that we have a big flush of seeds oftentimes. And then I had Emma, a great student that was helping to look and visualize all of the seeds to help us understand which ones were alive, which ones were dead, using the microscope and helping with all of the tetrazolium staining. So let's compare seed behavior at the soil surface to what I'm just showing you in that previous slide that's below ground. Here's the previous Sankey diagram that we were looking at before. So the major thing to remember is that the upper brown arrow is retained and viable seeds that are in the seed bank. We may find that some of those were actually dead, which is the gray. And then we have, in this particular case at the soil surface, a big green arrow, which shows the germination. And then there's some decay or unknown when we pull the seeds out. Decay and dead are very similar and will probably be combined in one category when we go to finalize all of these diagrams. So here's the same type of diagram, but now for buried seeds. I've grouped the data by core and edge region for seed source. And between the two Sankey diagrams, you can see there's a difference in germination and how it plays out. And it's very similar to what we saw in our seed burial experiment. So the green arrow in the soil surface diagram represents 85% germination rate, whereas in the diagram with buried seed, the green arrow is very thin and represents between 11 and 16% germination. When we look more closely at the core and edge populations, right now, we don't see that the percent of dormant seeds in the seed bank are very different from each other. They're quite similar. As we remove additional time units of seed bags in successive years, it will be very interesting to see if differences emerge between the core and edge populations as we move through time, and those seeds that are able to stay dormant persist. Those seeds that die from decay pathogens are disappear from this study, and then those that germinate are emerge from their seed casings. So this is a part of the study that I'm very excited to see what the future brings. So again, this is a 5-year ongoing study, but our preliminary results show that Stinkwort germinates after heavy rain events with high germination rates in the first year, but continued germination in successive years. Right now, that tracks what Rachel Brownsey found in previous research that she did looking at 3 years of seed bank data. Our study goes beyond 3 years as we're hoping to fill in where the dormancy might taper off through a 5-year study. We did see that germination rates are reduced when seeds are buried, and that was new and could potentially be helpful information for controlling. Finally, when we compare the initial results of this experiment looking at core versus edge populations, we see no difference in seed dormancy or viability. That was a lot to get through, and I appreciate you coming along for the ride. We've reviewed Stinkwort biology, and I've offered some research updates on how Stinkwort responds to disturbance and competition. And I've also given you a peek at some initial results from our seed bank study. Now, I'd like to take a moment to advertise a Stinkwort workshop that's coming up on Thursday, February 15th in San Jose, California, for anyone out there that's itching for more Stinkwort discussion. So this workshop is open for in-person attendance while seats are available, and there will also be some portions available online. A registration link will go live on our website later this month. And to give you an overview, the workshop is designed for local, regional, as well as state land managers, and also those researchers and concerned community members working with Stinkwort. The goals of the workshop are to disseminate information about Stinkwort biology, control, and monitoring techniques. We hope to facilitate networking, share best practices, and identify potential solutions for managing Stinkwort. Finally, I'd like to thank my collaborators and support system and funders, the United States Department of Agriculture, National Institute of Food and Agriculture, UC Santa Cruz Department of Ecology and Evolutionary Biology, UC Santa Cruz Doris Duke Conservation Scholars Program, the Center to Advanced Mentored Inquiry-Based Opportunities, UC Santa Cruz Plant Sciences Fund, and of course, the California Native Plant Society Marin Chapter. Thank you very much. If you have any questions about this talk, I can take them now, or you are welcome to email me or check out our lab website for more information on what we are researching. We keep it up to date, and there are many other invasive species that my lab looks at. If you are interested in the Stinkwort workshop, please copy down the tiny URL here or use the QR code with your phone or take a screenshot to navigate to a website where the registration link will go live later this month. I